Hello everyone, welcome to the channel Microbial World. In this video, we will be studying about Neisseria gonorrhoea, its morphology, cultural characteristics, biochemical characteristics, antigenic properties, pathogenesis, laboratory diagnosis, treatment and prophylaxis will be discussed. Neisseria gonorrhoea is commonly termed as gonococcus. In 1879, the gonococcus was first described by Neisser in gonorrheal pus. In 1885, Bum cultured the coccus and proved its pathogenicity. Members of the genus Neisseria dominate the mucous membranes of human and other animals. Neisseria gonorrhoea caused the sexually transmitted disease gonorrhea. Morphology. So, under the microscope, it appears as a gram-negative coccus which is present in pairs. Hence, it is called diplococci, with adjacent sides concave. The diameter ranges from 0.6 to 1 micromuum. The diplococci have kidney or coffee bean shape. It is a non-spore-forming bacteria and it is able to move using twitching motility. Okay, so the twitching motility will allow the bacteria to crawl along the surfaces. Okay, and the gonococcus possess pili on their surface, and it is the pili that facilitate adhesion of cocci to mucosal surfaces, and it also promotes virulence by inhibiting phagocytosis. Cultural characteristics. They are aerobic, means it needs oxygen to grow, but they may grow anaerobically also. Growth occurs best at pH 7.2 to 7.6 and at temperature of 35 to 36 degrees Celsius. It is essential to provide 5 to 10% carbon dioxide. They grow rapidly on chocolate agar and Mueller-Hinton agar. The selective media for Neisseria gonorrhoea is modified Thayer-Martin medium. Okay, so this medium, it is chocolate agar containing antimicrobials such as vancomycin, colistin and nystatin. Okay, and it inhibits most contamination including non-pathogenic Neisseria. The colonies formed on the media are small, round, translucent, convex or slightly ambonate, with finely granular surface and lobate margins. 4 types of colonies have been recognized, T1 to T4. Types 1 and 2 form small brown colonies. Type 3 and 4 form large granular non-pigmented colonies. T1 and T2 cocci are pileated, hence it is also known as P plus and P plus plus. It is auto-agglutinable and virulent. T3 and T4 cocci are non-pileated, hence it is also known as P minus. It forms smooth suspensions and are avirulent. So, fresh isolates from acute cases of gonorrhea, they generally form T1 and T2 colonies. On doing serial subculture, they will change to T3 or T4 colonial morphology. Okay? Biochemical characteristics. Neisseria gonorrhoea is oxidase positive because it has cytochrome C oxidase, and it is catalase positive, that means it can convert hydrogen peroxide to oxygen. Gonococci resemble meningococci except that they acidify only glucose and not maltose in serum sugar, whereas meningococci acidify both. It tests negative for nitrate reduction test. Rapid carbohydrate utilization test. This is a rapid sugar fermentation test that has now been recommended. And this test, it is based on the presence of preformed enzymes in bacteria and it is not dependent on the growth of bacteria in sugar media. It is rapid, more sensitive and specific test. Antigenic properties. The surface structure includes pili. Pili are hair-like appendages that reach out of the gonococcal surface up to several micrometers. They serve as virulence factors by facilitating attachment to host cells and inhibiting phagocytosis. It is composed of repeating subunits of protein called pilins. Pili undergoes antigenic and phase variations. Pore protein, which is also called protein 1. It is an essential outer membrane protein present on all gonococcal strains. They form transmembrane channels called porins. Okay, so these transmembrane channels, they play a role in exchange of molecules across the outer membrane. Okay, and there are two types of pore proteins, pore A and pore B, each with a range of antigenic variations. Only one type of pore is expressed by each strain of gonococcus. Okay. Oppa proteins, which is also called protein 2. These are opacity proteins that are variably expressed on gonococcal strains. These proteins are responsible for phenotypes of various colony. So the strains with oppa proteins, they form opaque colonies, and they are also responsible for attachment to host cells and clumping of cocci seen in urethral exudate smears. RMP, which is also called protein 3. These are highly conserved reduction modifiable proteins in the outer membrane. These proteins induce antibodies which inhibit the serum bactericidal activity against Neisseria gonorrhoeae. Lipo-oligosaccharide. This antigen has endotoxic activity, okay, and it is responsible for toxicity in gonococcal infections. Another protein is IgA protease. It splits and inactivates IgA. Pathogenesis. So as we said, gonorrhea is a sexually transmitted disease. Okay, so the disease is acquired by sexual contact. The first step in infection is adhesion of gonococci to the urethra or other mucosal surfaces. Pili is involved in this adhesion. The adhesion is rapid and firm. So what happens is that micturition, that is even urination after exposure, offers no protection against the infection. This is because the gonococci has firmly adhered to the surface. The cocci then penetrates through the intracellular spaces and reach the subepithelial connective tissue by the third day of infection. Then it penetrates between columnar epithelial cells. The period of incubation is 2 to 8 days. Gonococcal infection in men starts as acute urethritis with mucopurulent discharge containing gonococci in large numbers. Okay. So we know that urethra is the tube that carries urine from the bladder out of the body, right? So the inflammation of urethra is called urethritis. Mucopurulent discharge refers to discharge containing both mucus and pus. Okay. The infection then extends along urethra to the prostate, seminal vesicles and epididymis. Chronic urethritis may lead to stricture formation. That means stricture formation means it is the narrowing of the urethral tube. Okay. The infection may spread to periurethral tissues, causing abscesses and multiple discharging sinuses, water can perineum. Gonococcal infection in women. The initial infection involves the urethra and cervix uterine. Vaginal mucosa is not usually affected in adults. This is because the stratified squamous epithelium is resistant to infection by the cocci, and also because of the acid pH of vaginal secretions. Vulvovaginitis occurs in prepubertal girls. So what is vulvovaginitis? It is the infection or inflammation of the vulva and vagina. Okay, symptoms include like itching, redness, swelling and abnormal discharge and all. Okay. The infection may extend to Bartholin's glands, the endometrium and the fallopian tubes. Pelvic inflammatory disease and salpingitis may lead to sterility. Salpingitis means it is the inflammation of fallopian tube, okay, and pelvic inflammatory disease, it is an infection of the female reproductive organs including the uterus, fallopian tube and the ovaries. Okay, the symptoms can be pelvic pain, fever, abnormal vaginal discharge and also pain during intercourse. Okay. Rarely, peritonitis may develop perihepatic inflammation. From the word itself, you can understand it is the inflammation occurring around the liver, okay, and it is called Fitzhugh-Curtis syndrome. Disseminated gonococcal infection is a severe form of systemic illness. The characteristics of the disease include fever, arthralgia, that is joint pain which is caused by inflammation or injury, myalgia, which is muscle pain, migratory polyarthritis. Polyarthritis, it is the inflammation of multiple joints, okay, and rash. Proctitis occur in both sexes. Proctitis, it is the rectal lining inflammation, okay, and it develops by direct contagious spread in women and by anal sex in men. Conjunctivitis may occur by the auto-inoculation of patient's fingers. Blood invasion from primary site of infection leads to metastatic lesions such as arthritis, ulcerative carditis, and rarely meningitis. Ophthalmia neonatorum. It is a non-venereal infection in newborn. It results from direct infection during passage through the birth canal. Gonococcal bacteremia leads to skin lesions, especially hemorrhagic pustules and papules on the hands, forearm, feet and legs. It also causes tenosynovitis. Okay, it is the inflammation of synovium, that is the lining of the protective sheath surrounding the tendon. Okay, and this disease can cause pain, swelling, and also difficulty in moving the affected joint. Then it causes suppurative arthritis, which is infection of the joint. Okay. Laboratory diagnosis. Specimen. In case of men, purulent discharge or urethral swab is collected, and in case of women, urethral, cervical and rectal swab is collected. The morning drop of secretion may be examined. It may also be possible to demonstrate gonococci in centrifuged deposits of urine in cases where no urethral discharge is available. Microscopy. Upon gram staining, kidney-shaped gram-negative diplococci is observed. This provides evidence of gonorrhea in men. Diagnosis of gonorrhea by smear examination is unreliable in women. This is because some of the normal genital flora have similar morphology. The use of fluorescent antibody techniques for the identification of gonococci in smears has increased the sensitivity and specificity of diagnosis by microscopy. Culture. For culture, specimens should be inoculated immediately on collection. If this is not possible, then the specimen should be collected with charcoal impregnated swabs and sent to the laboratory in Stewart's transport medium. In acute gonorrhea, cultures can be obtained readily on chocolate agar or Mueller-Hinton agar incubated at 35 to 36 degrees Celsius under 5 to 10% carbon dioxide. In chronic cases where mixed infection is common and to examine the lesions, such as in case of proctitis, it is better to use selective medium such as Thayer-Martin medium. Growth is identified by morphology and biochemical reactions. Serology. Serological tests like complement fixation test, precipitation, passive agglutination, immunofluorescence and radioimmunoassay is carried out using whole cell lysate, pilus protein and lipopolysaccharide antigens. Molecular methods include PCR, that is polymerase chain reaction. Treatment. The Centers for Disease Control and Prevention, CDC, is currently promoting the use of ceftriaxone, cefixime, ciprofloxacin or ofloxacin as an initial treatment for uncomplicated cases of gonorrhea. For infections complicated by dual chlamydia, doxycycline or azithromycin should be added. Prophylaxis. Control of gonorrhea consists of early detection of cases, contact tracing, health education and other general measures. Vaccination has no place in prophylaxis. So this is all about Neisseria gonorrhea. Thank you.